The overall goal of the strategy is to generate an effective small molecule drug-like therapeutic through conversion of a peptide inhibitor of a protein-protein interaction. This is achieved by establishing a structure-activity relationship for the desired peptide-protein interaction to understand which parts can be effectively replaced. As a second step, computational chemistry is used to identify viable fragment alternatives for the known peptide determinants, which can then be used to synthesize a library of fragment peptide hybrids. Next, a library of hybrids is synthesized between these fragments and the truncated peptide in order to generate molecules that can be tested in an in vitro assay to identify molecules that are more drug-like and which recapitulate the activity of the native peptide. The results show more drug-like compounds can be identified using the methodology, and these represent effective starting points for drug discovery of inhibitors of protein-protein interactions.
So the main advantage of this technique over existing methods like high-throughput screening is that protein-protein interactions are extremely challenging from a drug discovery perspective, and conventional methods do not work well. The replace strategy involves a stepwise iterative approach and therefore may take longer, however is more likely to be effective in the long run. Today I will demonstrate most of the procedures myself with some assistance from my associate Dr. Sandra Craig, who will set up some of the reactions and procedures.